I'm Joe Greer, and I'm an assistant professor in psychology. And I specialize in clinical psychology, and we'll be talking a bit today about the lab techniques that we use in that subdivision of the field. Psychology is pretty broad and diverse, and so we have a variety of subdisciplines that range from neuroscience, cognitive science, social psychology, developmental psychology. And the labs within those subfields would all look pretty different. Certainly in neuroscience may be more akin to biology. In our labs in clinical or social and developmental, we're working with human subjects, which raises a whole other set of challenges than working with animal models. In my particular area, in clinical psychology, we have labs in two courses. In our 101 course, Psychology 101 is our introductory psychology course. And for that, we're introducing students to each subfield within psychology, and as a part of that introduction, they each participate in a lab. So I'll talk a bit about that lab, and then also we do labs in our 300 or advanced seminar courses, and I'll talk a bit about what those labs look like, as well as some of the challenges that go along with them. So in thinking about the intro psych course, as a new professor, I wanted to create a lab that was both interesting to students, an engaging topic, as well as introducing them to the scientific method within clinical psychology, and which is no easy feat within a 90-minute period. So the approach that I took to that is I wanted the students to have the experience of not only conducting a study, but being the participants in a study. So I had them come into the lab. It was a group of about 20 students, and I decided to focus the topic on an area that I thought would be personally relevant for any Williams student, which is about stress management. And I decided to have them make predictions about what happens to their physiological state during periods of stress, and then what happens to their physiological state during periods of relaxation. So in a sense, I was introducing them to aspects of the nervous system. I also wanted to introduce them to data gathering in clinical psychology. How do we conduct assessments? And then lastly, I wanted to show them a pretty standard type of clinical intervention that we use with patients in clinical settings for stress management. So what I did is I had students use a very simple biofeedback instrument, a little skin thermometer that measures peripheral skin temperature, and asked them to make a prediction about what would happen to their skin temperature in a moment of stress and then also in times of relaxation. This was a really great lab in the sense that over the course of 90 minutes, students were able to see what happens with their bodily functions as they became stressed. I told them that they each had to give a 2-minute presentation about something controversial in the field of clinical psychology, which they loved. And then I did a relaxation exercise with them subsequently, where I guided them through their entire body from head to toe, and I invited them to imagine the different parts of their body being calm and heavy and warm and relaxed. It's a very lovely type of relaxation exercise. Over the course of this, they recorded their temperature readings, and then we collected all those data, analyzed those data, and then by the end of the lab session, we were able to confirm or disconfirm their initial hypotheses about what would happen with their peripheral skin temperature under these different states. So in that way, it was very brief, and students were able to get a real-world view of what a clinical intervention is, how we collect data in clinical psychology, as well as experience what it's like to be a participant in a research study. At the 300 level, it can be a bit more challenging because now we're talking about a more advanced type of lab where we would, in my particular subdivision, we would really want to encourage students to think about the clinical applications of the research. And in clinical psychology, we're dealing with patient populations with different forms of mental illness or psychopathology. And so one of the natural challenges being at Williams would be how do we access these types of clinical populations? Now, even if we're unable to access populations, because sometimes it is prohibitive given the nature of a particular patient's problem, and we want to ensure that we protect human subjects, we will guide students through the process nonetheless of what it's like to propose a study and write up a proposal for an internal review board review of the scientific methods. And then we may do a version of that study that we can work with Williams students. So for example, on the topic of substance use, we may survey students about their substance practices on college campuses, which is relevant to clinical psychology, but also allows for us to access a population that's not necessarily clinical, but certainly is relevant to has relevant clinical applications. And we've done other things to find ways to access clinical populations. I had a wonderful student a couple years ago who accessed patients with diabetes via an online listserv, and was able to survey their eating patterns and the social influences on their eating patterns, which later turned out to be a wonderful study she presented those data at one of our annual meetings, one of our scientific meetings in clinical psychology. So with the wonderful creativity of the students, we do find ways to overcome some of those challenges, but that's probably one of the main ones that we do encounter. And as a result, often the lab sections in the advanced seminars tend to rely more on survey methods, or methods that students can access in terms of both populations and methodology here at Williams. So that's pretty much what we do.